Imagine a world where plants do more than just grow. They generate power. Our project explores a new frontier of symbiotic electrical power extraction from living plants and seeks to develop a sustainable, plant-friendly biobattery using biochemical energy instead of toxic metals. We aim to develop plant wearables, biodegradable energy harvesting devices that extract electricity from plants to power wireless sensors. These devices naturally degrade at the end of a plant's life cycle, making them environmentally friendly. And the key structure here is utilizing enzymatic biofuel cells implanted in plants. An enzyme biofuel cell is a bioelectrochemical device that facilitates the conversion of chemical energy into electrical energy through the use of biocatalysts. EBFCs use enzymes to drive redox reactions under mild and biocompatible conditions, and this property makes them particularly suitable for applications in biomedical implants, wearable sensors, and low-power portable devices. Research has found the use of EBFCs in humans and in animals, but has yet to find its place in plants. If possible, we could develop smart agricultural systems to facilitate autonomous energy harvesting and environmental monitoring, and this could enable real-time tracking of plant data, innovating precision agriculture. In addition, EBFCs have the potential to be a self-sufficient energy solution in a diverse set of environments. These all align with the sustainability development goals and will create a new age of sustainability and computing.
Through extensive literature research, we have found the average physiochemical breakdown of an opuntia or prickly pear cactus. We have found the amount and types of sugars and enzymes present in one plant under varying environments and growing conditions. This will enable us to conduct an EBFC experiment with real predictable enzyme contents and limits. Besides, we collect 24-hour sugar tests on our own opuntia. Through this result, first of all, we verify the sugar data of opuntia in previous literature review, and that also shows this plant conforms a typical chem cycle, which shows high sugar rate in daytime and low sugar rate at night. And these data will serve as a basis for subsequent chemical experiments. Based on the results found, our EBFC experiment will be conducted in varying amount of glucose solution to prove our design is also applicable to the Opuntia plant. In this experiment, we will use a Nafion membrane with two enzymes, glucose oxidation as anode material and lactose as the cathode material. We'll use gyroscope and pump sense to collect voltage data. This will give off a basis of how much energy we can expect for varying amount of glucose and enzymes. As mentioned, from our experimental design, we have picked out and purchased the materials we plan to use. We have found that glucose oxidase and lactose will act as the best anode and cathode respectively, and we chose carbon graphite paper for its conductivity and cost in comparison with carbon nanotubes. Lastly, we are using a Nafion membrane ferrocene and phosphorus citrate buffer, facilitates the transfer of electrons and balance the pH.
We also have found a few issues. One of the major challenges in our EBFC design is oxygen availability, as the oxygen levels inside the plant may be too low to support efficiency electron transfer. To address this, we are considering positioning the cathode near stomata or other air exposed plant surfaces to enhance oxygen supply. Using catalysts which can help store and slowly release oxygen is also a good way. Another issue is enzyme stability and storage conditions, as some enzymes, like glucose oxidase, require storage at -20 Celsius degrees, making them less practical for real-world applications. To overcome this, we are exploring alternative enzymes which can give a more stable chemical reaction and has a decreased oxygen requirement. Finally, the preparation of membrane and how to produce stable and visible electricity have always been a challenge to everyone. We will just try more and see how can we improve this. Our team plans to advance in our research on plant-based EBFCs and will soon conduct initial experiments to optimize their performance. If the findings are promising, we will design a secondary experiment to further refine our approach and enhance measurement accuracy. Once an effective solution is identified, we will evaluate energy output and longevity over a 6-month crop cycle, ensuring sustained power generation and real-world feasibility. Ultimately, our goal is to implant the EBFC system into living plants for smart agriculture monitoring.